Welcome to the deep dive. Today we're unlocking the secrets of a world that is profoundly familiar, yet, well, endlessly complex. We're talking about the world of flowering plants, angiosperms. From the food on our tables to the towering trees that shape our landscapes, these organisms are absolutely everywhere, and their internal lives are, frankly, a constant marvel. To guide us, we're drawing from a chapter of Raven Biology of Plants, the 8th edition. It's really a foundational text that brilliantly unpacks the diversity, structure, and unique life cycle of these dominant plants.
Right. And our mission for this deep dive is really to distill the absolute most important insights from this material. We want to give you a clear, concise understanding of what makes angiosperms just so successful. From the tiniest duckweed to the tallest eucalyptus. Think of it as a shortcut, maybe, to appreciating the hidden genius of their flowers and seeds without needing that textbook right in front of you. We'll be walking through the concepts pretty much as they unfold in the chapter.
Yeah, making sure to explain the processes in a way that hopefully truly connects. Okay, so when we talk about angiosperms, what truly puts their success into perspective? Like, what's the most jaw-dropping statistic that highlights just how dominant they are?
Oh, it's their sheer numbers, for sure. The phylum Anthophyta, that's the formal name for angiosperms, it boasts at least 300,000 species, potentially up to 450,000.
Whoa.
Yeah, that makes them by far the largest phylum of photosynthetic organisms on Earth. It's incredible. And to grasp their dominance, just imagine comparing, say, a colossal eucalyptus tree, like the mountain ash, right? Soaring over 100 meters tall, trunk almost 20 meters around.
Huge.
Huge. Compare that next to a duckweed, barely a millimeter long, just floating on a pond.
Yeah.
Both are angiosperms. And that incredible range really showcases their adaptability. You find them as climbing vines in rainforests, epiphytes living up in tree canopies, or, cacti thriving in harsh deserts. For over 100 million years, they've truly shaped pretty much every terrestrial ecosystem.
That's a really powerful overview of their incredible diversity. But to truly understand their success, we have to talk about what defines them. What are those sort of calling cards that set angiosperms apart in the plant kingdom?
Well, it really boils down to three key evolutionary innovations: their flowers, their fruits, and a truly distinctive life cycle. These features are so central that they unite all angiosperms into a single monophyletic group, which just means they all share a common ancestor who first developed these characteristics.
Okay, that makes sense. And within this massive group, we often hear about two major divisions, Monocots and Eudicots. Can you help us understand the fundamental differences between them and, like, why do those distinctions matter?
Absolutely. And they aren't just arbitrary classifications. They represent two wildly successful, yet quite distinct evolutionary blueprints. So monocots, think grasses, lilies, irises, orchids, palms, and even staple foods like rice and bananas. They typically have their flower parts in multiples of three. Their pollen usually has just one pore or furrow. They start life with a single embryonic leaf, that's the cotyledon, and their leaf veins often run parallel, like you see in a blade of grass. In their stems, the vascular bundles are sort of scattered around, and actual true woody growth is quite rare.
Right.
Now, eudicots, on the other hand, encompass most of the familiar trees and shrubs that aren't conifers, plus many common herbs. So think roses, oaks, sunflowers. They usually have flower parts in fours or fives, and their pollen typically have three pores or furrows.
Oh, okay.
They develop with two cotyledons. Their leaf veins form a branching, sort of net-like pattern, and their vascular bundles are neatly arranged in a ring within the stem. And unlike monocots, true secondary growth, what allows for thick woody trunks, is very common.
Got it.
So next time you look at, say, a blade of grass versus a rose bush, just remember these subtle differences reveal really distinct and highly effective strategies for thriving.
That's a really clear distinction, but it also makes me wonder, while most angiosperms are busy photosynthesizing, you mentioned some have taken a fascinating, maybe surprising detour in how they get nutrition. What's that all about?
Yeah, it's really a testament to their adaptability. While the vast majority are, free-living and photosynthetic, some have evolved truly unique strategies. We see parasitic plants like mistletoes or dodder. They develop these specialized structures called haustoria.
Haustoria.
They basically penetrate a host plant's tissues to, steal water and nutrients.
Wow.
And then there are myco-heterotrophic plants, like the Indian pipe. You might have seen it. It's ghostly white. It completely lacks chlorophyll.
Right, no green at all.
Exactly. They don't photosynthesize. Instead, they form this obligate relationship with mycorrhizal fungi. And these fungi, in turn, are connected to other green photosynthetic plants.
So the fungus is like a bridge.
Precisely. The fungus acts as a biological bridge, transferring carbohydrates from a green plant to the non-photosynthetic Indian pipe. It's a remarkable example of nature's ingenious and sometimes, slightly underhanded solutions for survival.
That's a real biological heist. Okay, but let's turn our attention to the star of the show.
Yeah.
The flower itself. What exactly is a flower from a botanical perspective, and what does the name angiosperm tell us about it?
Okay, so the flower is, in essence, what we call a determinate shoot.
Determinate shoot.
Yeah, meaning its growth is limited, unlike a regular branch,
Mm.
and it's specialized to bear sporophylls. These are basically modified leaves that carry the sporangia where spores are produced. And the name angiosperm itself, it comes from the Greek angine for vessel and sperma for seed.
Ah, vessel seed.
Exactly. This refers directly to the carpel, which is the flower's defining structure. The carpel is the protective vessel that encloses the ovules, those later become seeds, and its wall develops into the fruit. It's kind of evolution's ultimate marketing genius and reproductive machine, all rolled into one package.
And flowers aren't always just solitary blooms, right? They often group together in these fascinating arrangements. What's the benefit of these clusters, and how do botanists categorize them?
Right, they often cluster into what we call inflorescences. Think of the tight, dense head of a sunflower, or the airy, branched stray of a panicle, like on oats, or maybe the delicate umbrella-like clusters you see on dill or carrots.
Okay.
Each arrangement is really a strategic choice for optimal pollination and seed dispersal. It presents a more attractive or efficient target for pollinators, essentially. And just some terms here. The main stalk of a solitary flower or an entire inflorescence is called the peduncle.
Peduncle.
Yep. And individual flowers within that cluster often have their own little stalk, the pedicel.
Pedicel. Got it.
And all the flower parts then attach to a swollen tip called the receptacle. Now, to understand this masterpiece of reproduction, we break the flower down into its sterile and fertile parts. They're usually arranged in these concentric circles or whorls.
Whorls, right.
So the sterile parts form what's called the perianth. The outermost whorl is the calyx, which is made up of individual sepals. These are often green and kind of tough, protective, like the little green leaves at the base of a rosebud.
Okay.
Inside that, we have the corolla, composed of petals. These are usually the brightly colored, often fragrant parts, specifically designed to attract pollinators.
Mm.
Sometimes, though, like in lilies or tulips, the sepals and petals look so similar we just call them tepals.
Tepals. Okay, so the perianth does the advertising. What about the fertile parts, the reproductive core of the flower?
Right, moving inwards, we find the fertile parts. The male components are the stamens. Collectively, they're called the endoecium. Each stamen usually has a slender stalk, the filament, supporting the anther. The anther is typically a two-lobed structure, and inside it has 4 pollen sacs where the pollen grains develop. That 4-sac structure is pretty key for angiosperms.
4 pollen sacs. Okay.
Then we have the female parts, the carpels, forming the gynoecium. These are essentially modified leaves, kind of folded lengthwise, to enclose one or more ovules. Sometimes a single carpel, or maybe a group of carpels that are fused together, is also referred to as a pistil.
Pistil, right. I've had that term.
Yeah, that's
Yeah.
common. And each carpel, or fused pistil, typically has 3 distinct parts. The swollen base is the ovary, which crucially encloses the ovules.
Okay.
Extending upwards is the style, a stalk-like structure that acts as a pathway for pollen tubes.
Mm-hmm.
And the very top is the stigma. This is often sticky or feathery, designed to effectively capture pollen. Within the ovary, there are chambers called locules that house the ovules. And the way these ovules are attached inside is called placentation.
Placentation. That sounds like a very specific architectural detail. Why is there such varied internal architecture for seed attachment? What does it tell us?
Well, it's more than just aesthetics. It's actually a critical evolutionary clue. These variations, like ovules attached on the ovary wall, that's parietal placentation, or maybe on a central column in a partitioned ovary, that's axial placentation, these provide fundamental insights. They tell us about a plant's evolutionary relationships and how different species adapted to best protect and, disperse their precious cargo. Each type is really a strategic choice for reproductive success.
Fascinating. So beyond these internal structures, flowers vary outwardly too, right? In terms of completeness and sexuality. Can you explain those variations and what they imply?
Certainly, a flower is considered complete if it has all 4 basic whorls: sepals, petals, stamens, and carpels. Pretty straightforward.
Yeah.
If any of those are missing, it's incomplete.
Okay.
Then there's sexuality. A perfect flower is bisexual, meaning it has both stamens and carpels, functional ones, that is.
Both parts.
Both parts. An imperfect flower is unisexual. It has only stamens, we call that staminate, or only carpels carpellate. Now, here's a key point. An imperfect flower is always incomplete because it's missing a whole whorl of reproductive
Oh,
parts.
right. Makes sense.
But an incomplete flower isn't always imperfect. It could just be missing, say, petals, but still have both stamens and carpels.
Got it.
And this also extends to the entire plant. Monoecious species, like corn or oak trees, actually bear both staminate and carpellate flowers on the same individual plant.
Mm-hmm.
Dioecious species, like willows or hemp, have separate male and female plants, so you need two different individuals for full reproduction to occur.
Okay.
We also see parts fused together sometimes. Carination is when parts within the same whorl join up, like sepals fused into a tube. Adnation is when parts from different whorls join, like stamens fused to the petals. You see that in things like snapdragons or mint.
And what about the ovaries position relative to everything else? Or a flower symmetry? How do those characteristics factor in?
Right, ovary position is another key descriptor. In a superior ovary, which is common in things like lilies or tomatoes, the sepals, petals, and stamens are attached below the ovary. You can sort of picture the ovary sitting clearly on top.
Okay.
With an inferior ovary, like in apples or cucumbers, those other parts appear to arise from the top of the ovary. Think about the little dried flower remnants at the bottom end of an apple. That's a clue it had an inferior ovary.
Ah, I never knew that's what that was.
Yeah, and there's also an intermediate condition called perigynous, like in cherries, where a cup-shaped tube, called a hypanthium, forms around the base of a superior ovary, and the petals and stamens attached to that rim.
Okay, complex. And symmetry.
Symmetry, right.
Right.
Radially symmetrical flowers, also called actinomorphic or regular,
Yeah.
are like spokes on a wheel. Think roses, tulips. You can cut them into equal halves through multiple planes passing through the center.
Like a pot.
Exactly. Bilaterally symmetrical flowers, or zygomorphic or irregular, only have one plane of symmetry. Like snapdragons, orchids, or peas, you can only cut them into two mirror image halves in one specific way. And these variations in structure, position, symmetry, they're often really finely tuned adaptations to specific pollinators. They tell a whole story about the plant's evolutionary interactions.
It's clear the flower is just this marvel of evolutionary engineering, but let's dive into the core of their biology, with the life cycle itself. What's so unique about the angiosperm life cycle, especially compared to, say, other plants like gymnosperms?
What's truly fascinating here, and a key difference, is how incredibly reduced their gamophytes are.
Reduced, meaning small.
Very small, yes. The mature male gamophyte, which is essentially the pollen grain at maturity, typically consists of just 3 cells.
3 cells.
3 cells. And the mature female gamophyte, the embryo sac, usually only has 7 cells with 8 nuclei, and it's entirely retained within the ovule. This extreme miniaturization is a major evolutionary leap. It allows for much faster reproduction compared to, say, gymnosperms, and it leads to this indirect form of pollination.
So no more swimming sperm needing water like in ferns or mosses. Pollen gets deposited on the stigma, and then a pollen tube grows down to deliver the sperm. And this sets off that whole cascade. Ovule becomes seed, ovary becomes fruit. Can you walk us through how the sperm and egg actually form, maybe focusing on the key players, not every single microscopic step?
Absolutely. Let's start with sperm formation. It all happens inside the anther's pollen sacs. You start with diploid cells called microsporocytes or pollen mother cells. These undergo meiosis, that special type of cell division that halves the chromosome number to produce a group of 4 haploid microspores.
Okay, meiosis gives 4 haploid microspores.
Right. Each of these microspores then develops into a pollen grain. It does this by dividing mitotically, just once, forming 2 cells within the original microspore wall. This is the immature male ganophyte. You get a large vegetative cell, sometimes called a tube cell, and a smaller generative cell nestled inside it.
Vegetative and generative.
Exactly. Now that generative cell then divides again by mitosis to form the 2 male gametes, the sperm. This division can happen either before the pollen is even released from the anther or later while the pollen tube is growing down the style. So the mature male gatophyte is typically that 3-celled structure, the vegetative cell nucleus and the 2 sperm cells. And the pollen grain itself is remarkable. It's got that tough outer wall, the exene, made of sporopollenin.
Sporopollenin, right?
Yeah.
You mentioned that. Super tough stuff.
Incredibly resistant stuff. Protects against UV, dehydration, even pathogens. It's why pollen preserves so well in fossils. Inside that is a thinner wall, the intine. And don't forget the tapetum, that innermost layer of the pollen sac wall. It's super important for nutrition, and it secretes proteins and lipids onto the pollen surface, forming a unique pollen coat. This coat can be involved in recognition, scent, pigments, All sorts of things.
Okay, so that's the male side. What about the female side? What's happening inside the ovule to produce the egg cell?
Right, inside the ovule, within a central tissue called the nucellus, that's the megasporangium, a single diploid cell, the megasporocyte, gets singled out.
One cell.
Just one, usually. It undergoes meiosis to produce 4 haploid megaspores, typically arranged in a line.
Like the male side, meiosis first.
Correct. But here's the difference. Usually, 3 of these megaspores just disintegrate. The one that survives, often the one farthest from the ovule's opening, the micropyle, is the functional megaspore.
Okay, so only one makes it.
Typically, yes. This functional megaspore then undergoes 3 rounds of mitotic division without any cell walls forming initially. So you go from 1 nucleus to 2, then 4, then 8 haploid nuclei, all within the original megaspore's cytoplasm.
8 nuclei.
8 nuclei. These then arrange themselves. 4 move to the micropylar end, 4 to the opposite end, the chalazal end. Then 1 nucleus from each group of 4 migrates to the center. These become the 2 polar nuclei.
Polar nuclei, got it.
The 3 nuclei remaining at the micropylar end organize into cells. One becomes the crucial egg cell, and the other 2 become synergids. These synergids have a special structure called the filiform apparatus that helps guide the pollen tube.
Okay, egg and 2 synergids at one end.
Right, and the 3 nuclei at the chalazal end become the antipodal cells. Their function is less clear, often they degenerate. So the whole structure, the 3 antipodals, the 2 synergids, the egg cell, and that large central cell containing the 2 polar nuclei, that's the mature female gamophyte, or embryo sac. Typically 7 cells, 8 nuclei.
That cellular ballet is just remarkable. And you mentioned earlier that not all angiosperms follow this exact standard blueprint. What did recent research reveal about the really old lineages?
Yeah, that's a fascinating point. While this polygonum-type embryo sac we just described is the most common, found in about 70% of angiosperms, research, especially using molecular data, has shown it's not universal. Some groups, like lilies, have variations where, for instance, all 4 megaspore nuclei might participate, leading to weird ploidy levels like triploid nuclei even before fertilization. But even more interestingly, when we look at the most ancient basal angiosperm lineages, groups like Amborella, nympheales, water lilies, and Austrobaileyales, they have different setups.
Really? How different?
Well, water lilies and Austrobaileyales often have a simpler 4-celled, 4-nucleate embryo sac, and Amborella, which might be the sister group to all other living angiosperms, has an 8-celled, 9-nucleate structure.
9 nuclei.
9 nuclei. It actually has 3 synergids. These discoveries have really changed our understanding of early angiosperm evolution. It shows there was more experimentation early on than we maybe realized.
Wow. Okay, so this is where it gets really interesting because all these intricate developments set the stage for what you called the angiosperm superpower. Double fertilization. What exactly is double fertilization and why is it such a huge evolutionary advantage?
Right, double fertilization is precisely that, a superpower. It's almost unique to flowering plants, though something similar happens in netlike, but with a different outcome. So once viable pollen lands on a compatible stigma, it hydrates and germinates. It forms a pollen tube.
Right.
If the generative cell hadn't divided yet, it does so now, producing the 2 sperm cells. The pollen tube, carrying these 2 sperm and the vegetative nucleus, then grows down through the style. The stigma and style tissues are specialized to help this. Wet stigmas have secretions, dry ones have a cuticle, the style has transmitting tissue, all guiding the pollen tube.
So it's not just a passive journey.
Not at all. It's actively guided by chemical signals. And these tubes grow incredibly fast, much faster than in gymnosperms. They often deposit plugs of callus, a polysaccharide, behind the growing tip to maintain pressure. The tube eventually reaches the ovule, usually enters through the micropyle, and is guided towards the egg apparatus, likely by signals from the synergids.
Okay, the tube arrives.
It penetrates one of the synergids and then discharges its contents, the 2 sperm cells.
Right.
And here's the double part. 1 sperm nucleus fuses with the egg nucleus. This forms the diploid 2-end zygote, which of course will develop into the embryo.
Fertilization 1.
Fertilization 1, but that's only half the story. The other sperm nucleus migrates to the central cell and fuses with the polar nuclei.
Ah, the polar nuclei.
Exactly. Now, in the common polygonum type, there are 2 polar nuclei, N plus N. So this fusion involves 3 haploid nuclei, sperm N1 plus polar nucleus, and it's a triple fusion, resulting in a primary endosperm nucleus that is triploid, 3N.
Triple fusion. Wow. Okay, so that's fertilization 2.
That's fertilization 2. And this double fertilization is incredibly efficient. Why? Because it ensures that the nutrient tissue for the embryo, the endosperm, which develops from that primary endosperm nucleus, is only formed if fertilization of the egg actually occurs.
Ah, no wasted resources.
Precisely. It directly leads to the formation of both the embryo and its primary food source simultaneously within the developing seed. It ensures the embryo has immediate access to nourishment as it grows. That's a major, major reason for angiosperm success.
So this double fertilization event kicks off this remarkable cascade of development. What does it all mean for the plant? How do we get from that fertilized ovule to the seed and then the fruit we actually recognize?
It's a beautifully coordinated transformation, really, following double fertilization. First, that primary endosperm nucleus divides repeatedly, either with or without initial cell wall formation to form the endosperm. That's the embryo's vital lunchbox, packed with stored food like starch, oils, or proteins.
Okay, food supply first.
Often, yeah. Second, the zygote begins to divide and differentiate, developing into the embryo itself, with its rudimentary root, stem, and cotyledon.
Embryo develops.
Third, the ovule's protective layers, the integuments, mature and harden into the protective seed coat.
Seed coat forms.
And fourth, crucially, the ovary wall, the pericarp, along with sometimes other flower parts like the receptacle, starts to develop into the fruit that encloses the seed or seeds.
And that's the fruit.
That's the fruit. The endosperm is just so critical, providing that essential food for the developing embryo, and often for the young seedling when it germinates. And remember, this stored food, the endosperm, is formed after fertilization. That's a key difference from gymnosperms, where the food supply is the female gametophyte tissue, formed before fertilization. It's arguably more efficient.
Makes sense.
And that ovary wall, the pericarp, often thickens and differentiates into distinct layers, an outer exocarp, the skin, a middle mesocarp, the fleshy part, and an inner endocarp, like the pit around a peach seed. This gives us the huge variety of foods we see, from dry pods to fleshy berries.
Right.
It's truly a comprehensive system. It ensures the plant's entire life cycle, from that germinating seed to a mature flowering plant, producing its own flowers, achieving pollination and fertilization, and then packaging the next generation in seeds within fruits. It's all elegantly connected.
Before we wrap up, there's actually a surprising real-world connection in the chapter that shows how these tiny pollen grains can have a pretty big impact on us. You probably know it as hay fever.
Yeah, it's quite common. It's estimated that maybe 10 to 18% of people in the temperate northern hemisphere suffer from hay fever or allergic rhinitis, and it can be really debilitating for some.
So what's actually causing it?
Well, the culprits are often specific proteins found within or on the pollen grain walls. When these pollen grains land on moist surfaces, like our nasal passages, they release these proteins. And for susceptible people, these proteins act as powerful allergens, triggering that whole immune system cascade: sneezing, runny nose, itchy eyes, and wind-borne pollen, particularly from things like grasses, birch trees, and especially ragweed, tends to be a major irritant. Why? Because it's shed in absolutely vast quantities directly into the air. It just increases the odds of exposure.
Makes sense. More pollen, more problems.
Generally, yes. Although, interestingly, some plants that produce huge amounts of wind-borne pollen, like corn or pine trees, rarely cause allergies. So quantity isn't the only factor. The specific proteins matter a lot. Scent can sometimes play a role, too.
And for those of us in temperate North America, the hay fever season typically follows a pattern, right?
It does. Generally, you see tree pollen dominating in the spring, then grass pollen takes over in the summer, and then ragweed and some other late-season grasses are the main culprits in the fall. Of course, individual susceptibility varies wildly.
Yeah.
What's also, quote, notable, and maybe a bit concerning, is that the incidence of hay fever seems to have been rising steadily for, well, over 60 years now. And this is happening even in some areas where overall pollen counts might be stable or even falling slightly.
That's interesting.
It suggests there are likely complex interactions going on, maybe involving pollution, changes in exposure, or shifts in our immune systems themselves. It's a good reminder that these microscopic plant structures, these tiny pollen grains, can have a really profound and widespread effect on human health, far beyond just ensuring plant reproduction.
What an incredible journey into the really intricate world of angiosperms. From their just astounding diversity spanning those giant trees down to minuscule duckweeds to the delicate, precise architecture of their flowers, and then diving into that complex cellular dance of double fertilization and seed formation, it truly is a constant marvel of evolution.
Absolutely. You know, the aha moments for me really come from understanding that the variations we see, things like the distinct monocot versus eudicot features, or those different placentation types inside the ovary, they're not just arbitrary details. They are finely tuned evolutionary strategies. And that unique mechanism of double fertilization
Yeah.
just so elegantly ensuring the creation of both the embryo and its food supply pretty much simultaneously. It really highlights the genius of flowering plant evolution.
It really does. And as we close this deep dive, here's maybe a final provocative thought for you, our listener, to ponder. Considering the incredible evolutionary success and the sheer complexity of angiosperms and this intricate life cycle we've just walked through today, what hidden secrets might still lie within the seemingly simple act of a flower blooming? Secrets still waiting for us to discover. Thank you so much for joining us on this deep dive.
And a warm thank you from all of us here at the Last Minute Lecture team.